Privilege to introduce Dr. Sanford Simon from the Rockefeller University for today's talk. He is a Gunther Global Professor and head of the Lab of Cellular Biophysics, and he is basically well known in the area, I would say, of basic basic biochemistry and cell biology. He received his training initially at Princeton and then did a PhD at New York University School of Medicine, and I think he started his postdoc at Rockefeller and has been at the Rockefeller since then, and been very, very successful, and became even more successful in 2014 with one of, a true milestone and groundbreaking paper in fibrolamellar cancer. And this is obviously something very close to our heart for different reasons, because it's a liver cancer and it's a rare cancer. So it's something that, we here at the NIH very, very interested in. And since 2014, he has been working on this childhood cancer and looking at single well-defined cancers as well, especially in this area. And his team has identified the driver, the oncogenic driver, the specific fusion. I'm sure he's going to hear about this, the transcriptome, proteome, phosphoproteome, and help to understand the pathogenesis. And more recently, also how we can potentially treat this really bad disease. So without further ado, I would like you to take over and talk about advancing translational work in a rare cancer, or lessons from fibrolamellar carcinoma.
Tim, thank you very much. Can everyone hear me?
Yes, it looks good, and looks good and sounds good.
Very good. I get worried sometimes with the Zoom talk that I can be 20 minutes into it and discover I've been muted the whole time. What I'm going to tell you about today is a very specific cancer, a liver cancer called fibrolamellar hepatocellular carcinoma. It's a primary liver cancer, mainly affects adolescents, young adults. Peak detection is around 21 years with patients anywhere from 5 to 40 years old. Relatively rare. Part of the problem in studying it is that patients have no underlying liver disease. There's a paucity of symptoms, which has made it hard to diagnose. There's a lack of serum tumor markers. No clear geographic predilection, not known associated with any risk factors. It goes by a variety of names: fibrolamellar hepatocellular carcinoma, fibrolamellar carcinoma. I will use them interchangeably. It's still codified as a variant of HCC, hepatocellular carcinoma, but as I'll show you, has a completely different molecular pathology and totally different drug response as well. As a result, there's no standard of treatment, so there are no systemic chemo or immunotherapies that have been found to be effective. So really, the only current management is surgical resection. So this was first identified in 1956 by these large hepatocytes, polygonal cells. They have very prominent nucleoli. They have pale inclusion bodies. It's most often recognized by these fiber spans here. And actually, can you see my mouse? I want to be sure I'm doing it on the right screen. Yes? No?
Yes.
Okay, great, wonderful. Known as sort of lamella. Histopathology, it's positive for CK7, CD68, but because it's really not very clear, there's no established standard of diagnosis with suggestion of many misdiagnoses. So when we started working on it, what was not known, is it one disease or many diseases? Is it genetically inherited, or is it a somatic mutation? So our approach was to take patient samples that were well-defined as fibrolamellar, and then do whole genome analysis, transcriptome, the RNA-seq, proteomics, and phosphoproteomics. We initially skipped the whole exome because I figured if there was something obvious that was wrong in this, it would have been found by then. So when we did our whole genome analysis, it was actually underwhelming, which is actually maybe not surprising given that this is mainly in kids, and that there were very low level of single nucleotide variants in the DNA. There were no obvious mutations in any of the known oncogenes. There's very low level of structural variants. So for example, no recurrent amplifications, indels, inversions. The various programs we used said that there were no deletions as well, but I'll return to that. So this was sort of frustrating. So we switched to analysis of the transcriptome, and we found that there were over 1,000 transcripts that changed significantly. And the pattern of transcription was pretty consistent from patient to patient. So for example, if you looked at collagen, so the color is the ratio of expression in the tumor relative to the adjacent non-transformed for the same patient. So there's certain collagens that are very highly increased here. Some of them have a rather impressive false discovery rate. And that actually was not surprising because those bands that give it its name were known to be made of collagen. Certain other transcripts, for example, the EGF receptor pathway was highly upregulated. These are 3 of the ligands. So for example, if you go through these ligands here, it's 2 to the 6th, 2 to the 5th. Again, a fairly low false discovery rate. Or to give a sense of how consistent it is, this is just examining a few different patients looking at the extent to which neuregulin-2, a ligand in the EGF receptor pathways, upregulated. And you can see comparing tumor to adjacent normal, it's 2 to the 4.49, 2 to the 4.42. It's pretty consistent patient to patient. So from going through the transcriptome, we noticed a number of interesting oncogenes are upregulated: the EGF receptor pathway, the Aurora A, Wnt pathway, PAC3. So we initially confirmed all of our results by doing qPCR. Basically, the qPCR recapitulated what we had seen at the RNA-seq. So then we decided to look at the protein level. And we found, for example, if you look at ERBB2, it's increased in the tumor compared to normal in every patient with the Western blot. Aurora A is increased in the tumor compared to the normal in every patient. We then repeated the same studies using mass spec just to quantify it. Basically, there was a very good correlation between the RNA-seq and the mass spec. But it didn't really tell us what was going on with the cancer. We were a little perplexed. We started looking at non-coding RNA. We looked at a series of long non-coding RNA. So some of these here, you can see they're up 2 to the 11th, 2 to the 10th, again, with very low false discovery rates, 10 to the minus 63. If you notice here, these in red are ones that are known to be upregulated in liver cancer. This actually stands for highly upregulated in liver cancer, yet these were not significant at all. So this was really fairly distinctive from hepatocellular carcinoma. We noticed quite a few microRNAs that were changed as well. And we confirmed, again, all the microRNAs, the long non-coding RNAs with qPCR, they were tightly correlated. And there are a number of interesting links in there. For example, miR-548P, a microRNA, was decreased in fibrolamellar, and its known target, Frizzle-10, which is a Wnt receptor, was actually upregulated in fibrolamellar. So we were wondering, is that a correlation or is it causation? So we tried doing was overexpressing an antisense to the miR, to the microRNA, and we found that the more we knocked down the microRNA, the more we increased Frizzle-10, or if we overexpressed the microRNA, then again, Frizzle-10 went down. So there seemed to be a link between these, but it wasn't really telling us, what was driving the pathogenesis or what were good targets for therapy. We noticed that the pattern of transcription clearly segregated away in the tumor from the adjacent normal in all patients. We also noticed that they were completely different from what we saw in any of the liver tumors. We looked at cholangiocarcinoma, hepatic adenoma. Here are some examples of hepatocellular carcinoma that we downloaded from dbGaP. All these hepatocellular carcinoma patients segregated away from the fibrolamellar, except for this one patient, which once we did sequencing, we discovered was in fact a misdiagnosis. We found over a dozen kinases that were upregulated in every patient, some of which are sort of known to drive certain tumors like Aurora A, the EGF receptor, the catalytic subunit protein kinase A, PAC3. But when we looked, there were no changes in the coding sequence. There were no changes in the promoters. So what was causing this difference in expression? And one thing we decided to look at was there changes in splicing. There's certain cancers, like certain breast cancers, you get a change of splicing, a change in the use of exon. And the way you can assess that is by taking all your RNA reads and mapping the RNA reads on the DNA. If there's a change of exon, what you should do is you should have reads on each exon, and you should get reads that span the exons when they got spliced together. We found for all these different oncogenes, all these different kinases, all these genes involved in liver development, the splicing was completely normal, with one and only one exception, the catalytic subunit of protein kinase A. So this is now on the negative strand. So I apologize, this is reading from right to left. We found increases in the transcriptome in exons 2, 3, 4 onward, increases in the reads between the exons, but no increases in the first exon. The first exon was at normal levels, and no increase in the reads between these two. Instead, there were reads that went up 400 kilobases to a heat shock protein, DNA JB1, to the first exon. And so these reads suggest we have the normal DNA JB1 being spliced together, the normal catalytic subunit protein kinase A, but now this chimera, this fusion of the first exon of DNA JB1 to the rest of PRKCA. And in fact, when we went into the liver tissue, we found this fusion in every single patient we checked for, in addition to the normal genes. So what's responsible for this? Is there an altered splicing or something else? So we went ahead and walked along the chromosome and found that every patient had a normal copy of this particular chromosome, chromosome 19. But on one copy of the chromosome, they had a deletion somewhere from the middle of the first intron of PKK, and usually the first intron in the middle of the second exon of DNA JB1 that would splice it together. We found this in 15 out of 15 patients. But once these get fused together, when you do the RNA splicing, it ends up fusing the first exon of DNA JB1 to the rest of the catalytic subunit of protein kinase A. So I've frequently made fusion transcripts in the lab for the purpose of studying certain proteins. I'll make a fusion to GFP or to other targets. And often these fusion transcripts don't express as a protein. So was a fusion protein being made? So when we looked in the adjacent non-transformed tissue with an antibody against PRKCA, we saw a single band. But when we looked in the adjacent tumor tissue, we found a slower moving band consistent with being this fusion protein. So was this really a true fusion protein? To test this, we used an antibody against the DNA JB1 to immunoprecipitate it, and then probe with an antibody against the opposite end, the catalytic subunit protein kinase A. And in fact, this slower moving band was a true fusion protein. It can be pulled down either with antibodies against DNA JB1 or with antibodies against PRKCA. In fact, when we looked in the tumor tissue, there was an increase in the amount of catalytic subunit relative to the adjacent normal, which we think is due to now the DNA JB1 promoter driving the transcription of this. When we purified either the native kinase or the fusion kinase and just measured its activity, sort of on a gross macroscopic level, it had the normal activity. So this was now found in every patient we tested. We've now tested roughly 350 samples from different patients. But this is a correlation. It doesn't tell you if it's really driving the tumor. So to do this, we got together with Scott Lowe's lab, who's across the street from us at Sloan Kettering, and we used CRISPR-Cas9 to engineer this deletion into the liver of mice. When we did this, we got tumors that looked to the pathologist like fibrolamellar. They had all the pale bodies and the altered nuclei. The transcriptome was the same as in fibrolamellar, and the mice would die of their tumors. But this particular experiment doesn't really distinguish whether it's the creation of this new chimeric fusion that's driving it, or maybe it's loss of the usual level of the heat shock protein or the catalytic subunit, or maybe it's the loss of those 8 genes that are in between. They have the same 8 genes in that locus in mice that we have in humans, or maybe it was the loss of a non-coding RNA in the intervening region. So to do that, we created this fusion gene and just expressed it on its own with a transposon in mice without doing a deletion. When we did this, we got tumors that, again, looked to the pathologist like fibrolamellar. They had all the proper histopathology. The mice were dying of the tumors, and they had all the transcriptome. What this approach allowed us to do then is address the following. Was fibrolamellar solely due to increased expression of PKA, the catalytic subunit, due to this driver, or was it something different going on? So we tried overexpressing just the wild type PRKC with the same driver. While this altered the cells, it did not produce a transformation. So there's something very specific about this kinase. Now, one thing about kinases is that they both work as a kinase on their own, adding a phosphate, but they also act as scaffolding, often joining together through the A kinase-associated proteins and through other phosphatases. They form a scaffolding function. Was the kinase activity itself actually needed? So we did this again. We transformed cells with this transposon, with the GFP. This is now in the liver of mice. And then we did it using a kinase, which had a single point mutation to kill the kinase activity. And again, no transformation. So this is, you really need the kinase activity. So now what we want to do is go forward. So these cells were expressing it, they just weren't transformed. So now how do you go about fighting fibrolamellar? To do this, what you really need is validated model systems. And I'll give you 3 of the systems we've been using in developing therapeutics. The first being organoids. I'm going to validate these organoids we've made from fibrolamellar patients. We have them also from the adjacent normal based on continued expression of this fusion kinase. We're going to validate it based on continued expression of the fusion protein here. It's running slower in the gel. But also, we're going to look at the transcriptome and look at the extent to which different transcripts increase or decrease in expression relative in the organoids relative to what they do in the patient tumors. That's one model system. Second model system, we're going to take tumors directly from patients, dissociate the cells, and screen those. And for this purpose, most of the work I'm going to show you has been done by getting a tumor that's been resected, and we'll go into the OR, take the samples right from the surgery, dissociate the cells, screen them over the next 72 hours. So we'll have a result within 72 hours of resection. Lately, we've been switching to doing the same thing, but with needle biopsies. The third test model system I'll show you is taking tumors and implanting them in immunocompromised mice. And again, these tumors will be validated, these patient-derived xenografts, based on do they recapitulate the histopathology in the PDXs relative to the original patient's tumor? Do they retain expression of this fusion oncokinase compared to the adjacent normal? And do they retain expression of the fusion protein compared to the adjacent normal? And then finally, do they recapitulate the transcriptome? So do transcripts that go up in the PDX also go up in the patient tumor compared to the normal? So this is our validation system for these. So now what we did is we took these PDXs, we dissociated the cells, separated out the human from the mouse cells, and we can now screen them against different drugs. And the first thing we did was to test for various drugs that are now actively in the clinic, fibrolamellar. For example, sorafenib is one of the most commonly used drugs. And in all cases, our counter screen is going to be here in black, primary human hepatocytes. And we saw no effect of sorafenib or any of these other drugs against fibrolamellar. Excuse me. So we were wondering, was this a problem with our test system? Maybe when you take the cells out of the context of the liver, out of the context of the mouse, maybe they just respond differently. So we start testing the same drugs growing in mice, same drugs on tumors growing in mice. And again, sorafenib had no effect on tumors growing in mice, nor did any of the other drugs. In fact, this was a collection as of 18 months ago. We've now tested many more of drugs that have been used in the clinic or implicated for use in the clinic in fibrolamellar. And none of them had any consistent effect at all. So we decided to take a more precision medicine-oriented response to it. I mentioned earlier we'd identified a few oncogenic pathways that were upregulated in fibrolamellar. Let's try to block those. So for example, we blocked the aurora kinase A pathway. Again, there was no effect on the fibrolamellar cells relative to using primary human hepatocytes. We tried blocking the EGF receptor pathway. Again, no effect. We've done the same thing with Wnt, with other pathways. So blocking oncogenic pathways that are upregulated had no effect on tumors either dissociated in cells, or for most of these, we've also tested them growing in mice. So the question is, how do you proceed therapeutically? There are a few strategies. You could hit the machinery that encodes this chimera, this fusion gene. You could hit the chimeric protein here. You can hit the immediate downstream elements that are activated by it, or you can activate the immune system. So to do these, you want to know, are any of these actually valid targets for therapeutics? So for example, the DNA JB1 Prokaka, we know it's sufficient to trigger fibrolamellar, but there are many examples of tumors. I know quite a few RAS tumors, for example, where they will trigger the tumor, but then if you eliminate that oncogenic trigger, the tumor continues to grow. So does this only trigger fibrolamellar, or does it continue to drive fibrolamellar? The second question is, if we knock it out, even if it's continuing to drive it, do the cells then normalize, or do they die? To me, that was very important, because if the cells just normalize, that means we have to continue to give this therapeutic the rest of the patient's lives, which allows for the potential for mutational escape. So the way we tested it was the following. We designed a series of shRNA that spanned the fusion junction. We initially tested this out in Hus7 cells, a HCC line which we had put in the fusion kinase, just to see can we probe for knocking down specifically the fusion protein, but not any of the parental proteins here. And we identified a few shRNA that were very promising. So then we went back into fibrolamellar cells and showed that there they also very successfully knocked down the fusion and had no effect on the parental proteins. So now the question is, do they have any effect on the growth of the tumor? So first we showed that in vitro, they actually killed the tumor. The critical things, what do they do in a tumor growing in a mouse? So we did is we had the shRNA be inducible inside the fibrolamellar cells. We implanted the tumors. We allowed the tumors to grow to the point that we can measure them. And then we induced the shRNA. And in all the different PDX lines that we tested, once we induced the shRNA, the tumors die. So this suggests that if you knock down the fusion transcript, which knocks down the fusion protein, the tumors die. However, there was one nagging doubt. It could be that when you form a double-stranded RNA, maybe it's activating some other cell distress pathway, maybe reactive oxygen species, maybe some of this is recognizing a double-stranded RNA. Is this really very specific to knocking down the fusion protein or the fusion transcript? To test this out, what we did is we put the fibrolamellar fusion gene into an HCC cell line, but we didn't select for it to become oncogenically addicted. We just transduced it in, allowed it to start growing in mice, but then induced the shRNA with doxycycline. And in that case, even though we knocked down the fusion transcript, there's no effect on growth of the tumor. So that really argues that it's specifically in fibrolamellar where you knock down the fusion transcript, you knock down the fusion protein, it's become oncogenically addicted to it. And to me, that validates this as a therapeutic target. So our strategies now are to hit the RNA, and you can see from what we're doing here, some of the directions we're going. But also the second strategy is to hit the protein. So the question is, how do you selectively hit the fusion protein? So we've got the crystal structure for both the original kinase, as well as the crystal structure here for the fusion protein. This here is the fusion domain added on. So it turns out this kinase is actually part of a holoenzyme where you've got the catalytic domain here together with two regulatory domains. So we did a high throughput screen of 500,000 compounds looking for compounds that would block the kinase activity. And from these, we came up with a series of compounds that blocked pretty well, meaning below 5 nanomole. This is actually work that we're doing with Ping Zhang at the NIH together with Ning Li, where we have the structure now of some of these bound in. But the problem we had is that if you look, this is the inhibition against the fusion kinase and the inhibition against the wild type kinase. It inhibits both. So actually, here's 4 of the crystal structures that Ping and Ning had generated. In all cases, inhibition 2.1 nanomole, 1.6 nanomole, it's inhibiting the wild type kinase over the same range as the fusion kinase. So the question is how to proceed. So we're going in 2 directions, one of which is to take advantage of these crystal structures to modify the basis of the compounds. But the second approach we're taking is the following. We have this structure. What we're doing is we're extending these small compounds with a linker attached to an E3 ligase to now ubiquitate the fusion kinase. So why are we doing that? It turns out that this fusion domain here has many, many lysines on it that are eligible to be ubiquinated. And the main the main kinase itself is very tightly folded. So it's not a good substrate for the proteasome. Whereas this domain here is very, very flexible, making it a good domain for the proteasome. So now we've made PROTACs with 6 of these targeting both and tested in both the wild type kinase and the fusion. And what we can show now is that when we induce this, it's called a PROTAC, right? So one end binds to our oncokinase, the other end has an E3 ligase. As we induce it, we can show we can selectively eliminate the fusion kinase, but not the wild type kinase. And if we induce it in certain select cells, we find that only in the cells where we induce it, we can knock out PRKCA and the cells die. So we're actively pursuing this now. We're doing tests on it in mice. We're now also looking for additional small molecule PROTACs. We've now screened 650 million compounds, identified 25 that uniquely bind to the chimera. And we're now fine-tuning their binding using SPR and getting different variants on it. So we now have success in a number of these approaches, both in vitro and in mice. The problem with this work, though, is that these are new agents. They're going to take time to go through all the different testing you need to bring them into the clinic. And people are ill now. So the question is, what can be brought to the clinic now? So in parallel, what we've been doing is a drug repurposing screen, in which, once again, we'll take these patient-derived xenografts, separate out the human cells, and do a screen on basically everything that's been safety tested in humans. So we started off with 5,100 drugs. Actually, we've now repeated this recently with an additional 3,000 on 2 of the PDX lines, and took our top 200 hits and did a dose response curve from 10 micromole all the way down to 10 nanomole. In all cases, our counter screen is going to be primary human hepatocytes. So here are the results against some of the top cells from the fibrolamellar screen. And there are a number of surprising hits on the fibrolamellar cells compared to the primary human hepatocytes. For example, there are certain drugs that affect marks of the chromatin. In fact, in general, there were a lot of HDAC inhibitors that we had. Irinotecan, this is a topoisomerase I inhibitor. This is something that's supposed to work on rapidly growing cells, but worked in fibrolamellar. navitoclax. It blocks BCL2 and BCL-XL, two proteins that are involved in cell death pathways. And along the way, we noticed that the response of the cells actually varied from line to line in some cases, like in SN38. We began to wonder, is it possible that expression, differential expression of some of these cell death proteins, these anti-apoptotic proteins, could be responsible for the variability in our response? So we decided to take certain specific tool compounds and see, do they synergize with panobinostat? Do they synergize with irinotecan and other drugs? And we found specifically drugs that block BCL-XL synergized, whereas drugs that block BCL2 did not. So another way to look at it, here's the response, dose response curve of 3 of the fibrolamellar lines against panobinostat. But if we block BCL-XL at the same time, you can see they're much more sensitive. Whereas if we just look at the primary human hepatocytes, they do not sensitize when you block BCL-XL. So we repeated these tests using quite a few of these drugs. They were all very successful in showing synergism. What I want to jump to right now is doing tests in mice. So here in blue is the tumor growing on its own. This is just using navitoclax, which blocks BCL2, BCL-XL. We use that because it actually has been in the clinic. Here's irinotecan, which is supposed to block rapidly growing tumor cells, which again was surprising because fibrolamellar is slow growing. And you see it stops the tumor growth. When we combine the two, we actually had tumor shrinkage in all of our models. At this point, we got a little worried for two reasons. First, we were concerned, how do we know the tumors grown in mice are really reporting on the bulk of the tumors in patients? So they recapitulate the transcriptome, and that's sort of a very macroscopic measurement. Maybe there's 1, 2, half a dozen transcripts that are different. When we put these tumors into mice, they'll often take 9 months before they take hold. Maybe we're actually subcloning only a small percentage of the cells. And what we're looking at is the drug response profile of those cells, not really the full population in the human patients. The second concern is that navitoclax, even though it was in the clinic, has adverse effects on platelets, and the manufacturer is actually pulling back on it. So the first question, how do we know the tumors are reporting the bulk of the behavior of the tumors in mice? So we started doing all of our tests on tumors directly dissociated from patients. We were looking at the entire population of tumor cells. So when we do that, you can see there are some cells that are sensitive to irinotecan and some that are hypersensitive relative to the primary human hepatocytes, some that are sensitive to panobinostat and some hypersensitive to panobinostat. So these are all cells taken directly from a resection. And in most cases, we started testing the drugs within an hour of resection. Again, there's difference in their sensitivity. And we were wondering, was this due to the expression of the anti-apoptotic proteins? So what we're going to do is on these cells directly from the patients, we'll take this patient sample. I'll show you this patient sample who's the least sensitive to irinotecan over here. And at the same time that we did this study, we also had parallel plates where we had increasing concentrations of navitoclax to block BCL2 and BCL-XL. And you can see here, when you get up to higher levels, we're driving the sensitivity to irinotecan down to the single-digit nanomolar range on a tumor that actually is not supposed to be responsive. So that answers the first problem. Is really recapitulating what's happening in the patient tumors. Now, what about the problem with navitoclax and its effects on platelets? Fortunately for us, another group had been developing a PROTAC. This is, again, one of these devices that will send your protein to the proteasome for destruction. But they did it by using an E3 ligase that's not found in platelets, a very low level in platelets. And when we tested it out in fibrolamellar cells, we found we could completely eliminate BCL-XL within about 24 hours. And fortunately, in the way they designed their PROTAC, it turns out it doesn't affect BCL2 at all. So it's really very selective for BCL-XL. And when we treated mice with it over time, we saw there are no adverse effects on the platelets from treating with this PROTAC, which is actually quite different from what we had with navitoclax. So now, will this work on tumor growth in mice? So in the upper panel, what you're going to see is the average of the different mice. Down below, I'll give examples of individual mice. And this is the treatment schedule. So irinotecan was given Monday to Friday, a weekend off, Monday to Friday, and then a full week off, Monday to Friday, Monday to Friday. And we did treatments here of 6 cycles. When we gave irinotecan, the tumors stopped growing. There are some cases was maybe a slight decrease. However, when we combine this together with the PROTAC to block BCL-XL, not only do tumors go down and completely disappear, once we stopped treatment and waited 8 weeks, the tumors did not come back. And we've done this now on 3 or 4 independently derived patient-derived xenografts. So the current status is that this PROTAC DT2216 is currently in phase 2 testing as a monotherapy for the various blood-based tumors. It turns out from going into the patient medical registry we have, which I'll go into in a moment, we actually found 3 patients who had been given irinotecan as a sort of agent of last resort. And in all 3 cases, what happened in the patients was the same as what we saw in mouse. The tumors essentially either stopped growing or there's a slight decrease in time. So currently we're working to develop irinotecan together with DT2216 as investigational drugs for fibrolamellar. But we were a little disturbed, like, why would irinotecan be working? It seems important to understand this if we want to be able to really choose our therapeutics. And a clue came from doing the proteome of the cells. So it turns out irinotecan is made into this active metabolite, SN38 that I referred to. And in the liver cells, the liver gets rid of it by putting on a sugar group on it to then export it out. And that's how they detoxify from irinotecan. It turns out that in fibrolamellar, when we did mass spec in the proteome, this family of enzymes is actually down quite significantly in the tumor cells. In fact, we found by going ahead and quantifying the level of this enzyme in different patients, as well as the level of BCL-XL, we can predict the drug synergism between irinotecan and blocking BCL-XL in these cells. And actually, from doing this, really pointed out to us some of the power of going after not just the transcriptome, but the proteome in these cells. So we started doing was getting patient samples in which we would do mass spec, and we've done mass spec both by doing tandem mass tag as well as doing label-free quantification. And when you do that on the patient samples compared to the adjacent normal, there's some proteins that will increase, some that will decrease, both by either technique. The results are essentially indistinguishable. There were also a lot of interesting changes in the phosphoproteome, some that increased and some that decreased in the fibrolamellar cells. And if you do a clustering analysis, what you find is that in general, the tumors from the fibrolamellar tissue segregate completely away from the normal tissue. And here's the tumor, the normal. Same thing, the phosphoproteome segregates away between the tumor and normal. I specifically left in this set of patients for a specific reason. There was one patient that was an anomaly, patient number 4, where the tumor proteome segregated together with the normal for all the other patients, and the phosphoproteome also segregated away with the normal. So the way we usually test to see if a tumor is fibrolamellar is by doing a PCR reaction between the first and second exons of PRKCA to see, do they have the normal kinase, and a PCR reaction between the first exon of DNAJB1 and the second exon of PRKCA to see, do they have the fusion transcript. And when you do that, what you see usually in the tumor, you get a band for this fusion gene, a band for the wild type. In this case, from this particular patient, we also had a lymph node that we tested on. And the adjacent non-transformed tissue, if it's a clean resection, we don't get a signal for the chimera. It turns out for patient 4, when we probed for it, we were never able to get a band for the fusion protein, for the fusion transcript. I repeated this over and over. So at this point, we took 300 of our H&E samples and took away the labels, and we sent them blind to a pathologist at Mayo Clinic, Mike Torbenson, and told him, okay, go through all of these samples. And he came back and everything that we had, and he had them blind. Every case where we had seen a fusion transcript, he said it was fibrolamellar. He identified the normal. This particular patient he identified as being hepatic adenoma, and therefore not fibrolamellar. So from going through looking at the transcript, looking at the proteome and the phosphoproteome, there were some proteins. So this is the normal on the left. This is the tumor on the right. The black dots are the protein level, and these lines here are particular phosphocytes. We found some proteins would increase a protein level, and the phosphorylation increased at all sites. Some proteins like this, the protein level didn't increase, but the phosphorylation at this site increased quite significantly. Other proteins like Cathepsin-2, the protein level actually went down, but phosphorylation went up. So there's something strange going on with the phosphorylation. So we decided to see, was it something very special about this fusion kinase or not? Was it the same as the wild type? So approach was to take human liver, isolate the cytosol, kill all background kinases there, and then take the inactivated kinase, cytosol, and add into it either the wild type kinase or the fusion kinase, and then ask what proteins get phosphorylated and do mass spec on those. And when we did that, we found there were a lot of phosphocytes that were shared between the fusion and the wild type kinase, but there's some proteins that were uniquely phosphorylated by the wild type, by the fusion kinase here. This is true whether we did label-free quantification. It was also true from tandem mass tag. There were certain things that were phosphorylated by the fusion and not phosphorylated by the wild type, and other things that were phosphorylated by both. And when we looked much more closely what was being phosphorylated, we saw they sort of shared the same basic motif with the following exception. The fusion kinase was actually phosphorylating things that had a proline in the +1 position, and also like to have a negative charge here in the +2, +3 position. And that seemed to be very strange because usually PRKCA, protein kinase doesn't phosphorylate things with a proline in the +1 position. From looking at our MD simulations, we discovered while the pockets are almost identical, there's enough of a subtle difference that actually the fusion can actually accommodate in the proline in the +1 position, which is what we think is giving these new phosphopeptides. So from looking at, in general, the increased proteins and the phosphoproteins, one thing that was very striking is that there were a large number of them that were in the mitochondria. These are ones that were affecting either ammonia metabolism, mitochondrial biogenesis, or metabolism. And it's particularly caught our attention because one of the frequent causes of death in fibrolamellar patients is high levels of ammonia. And these patients do not respond to classic treatments like lactulose to sort of shunt past the liver. So we had the results of the mass spec were very compelling, but we decided to check again doing immunofluorescence. And we found, we looked, for example, at an ornithine aminotransferase that said it was increased, or PYCR1 or PROD-H. We found they were increased not in a subset of cells, but it was increased in all of the tumor cells. So the increase was not in the stromal cells, but in every one of the tumor cells. So what's going on? What is this telling us? Well, one thing we noted is that a lot of the changes were in two pathways. This pathway here is responsible for detoxification of 35% of the ammonia in the body. So ammonia is made into carbonyl phosphate, which is then taken in by ornithine transcarboxylase, joins it together with ornithine to feed it into the urea acid cycle. This pathway of glutamate going to glutamine is responsible for another 35%. So 70% of the total ammonia detoxification in the body. This enzyme is down quite significantly, 2 to the 5.54. And this enzyme is also down. And not only is this enzyme down, but because ornithine aminotransferase is up, it's sucking away a lot of the ornithine. So this is just showing the proteins are up. What we really care about, though, is what about the substrates of the products? Are they really being altered? So to do that, we looked at the metabolome, and there are certain particular predictions you'd make. So it turns out there are patients who have genetic mutations in OTC, so they have very low levels of OTC. And these patients very early on in life present not only with high ammonia, but because they can't consume the carbonyl phosphate, this leaches out of the liver, where you can start seeing the metabolites, carbonyl aspartate, dihydroorate, and orotic acid. So when we did the mass spec on the tumor compared to the adjacent normal, the three top metabolites we found increased were in fact these that you'd expect to get if you had a block in this particular pathway. We also found, for example, in this pathway here, that glutamine was actually down quite significantly, which you'd expect if the GLUL is down. But also we noticed that this pathway, that actually the reverse pathway, was also quite significantly increased. And what this data is suggesting is that not only are you compromising the ability to consume ammonia here, you're compromised the ability to consume ammonia here, but actually you may actually be getting production of ammonia by this now running in reverse over here. So to test this out, we did a few things. We partnered with the patients who have a medical registry. It turns out a number of their patients had measurements of orotic acid in the urine. The normal range is 0.2 to roughly 1 millimole of creatine. In the patients, it was roughly 1,000-fold increased for the patients who had fibrolamellar. So one thing we're actually starting to do now is use orotic acid as a diagnostic for some of these patients. But the other thing we noticed was the following. This is predicting you're actually getting production of ammonia. So we found a collection of 8 patients who had had surgical resection of fibrolamellar. It was basically a debulking reaction. And in all the patients, immediately upon debulking, there was a decrease of the ammonia into circulation. And going through the literature, there are quite a few other papers that support the same point. So actually, it's suggesting there's not only a compromising consumption of ammonia, but ammonia production, which gives suggestions for therapy. So from partnering with the patients, they've put together a medical registry. We have over 200 patients now. The advantage is just a limited number of patients. There's only so much you can do with existing data. This way, we can also follow the patients over time as they move from institution to institution, even across countries. So for example, we have many more patients who signed up, but we found roughly 10% of these over time, we discovered they did not have fibrolamellar, so we were able to remove their data from the registry. It's run by the patients, so the patients feel comfortable that the data is never going to be sold to companies. So we have a fairly high buy-in. And we've also asked them all sorts of additional questions, not part of the regular electronic medical record, which has actually given some interesting insights. We're trying to provide a link between the patients and the clinicians and the science communities. We give them frequently asked questions about fibrolamellar. We have definitions of various terms. We have a whole video on how to go into PubMed, how to search PubMed. We have plain language summaries for a lot of the articles that have been written about fibrolamellar. And actually, for a few of the cases, we've actually made little videos that describe the papers. So,
in order to confirm that the chimera actually causes fibrolamellar, the researchers edited the genes of adult mice using the new CRISPR technology. They took the DNA of adult mice and created the same DNA deletion found in fibrolamellar patients.
So we tried to present the science in a form that was accessible, but a way they could follow. The whole registry is IRB approved. We have various epidemiologist decisions going through it. For example, Mark Yarshu and Hopkins just had a paper come online this week where he collected data from many of the patients in the registry. So more than half of his data comes from there who'd had immune checkpoint inhibitors. Various epidemiologist statisticians are going through. So for example, there are clusters of different patients in different locations, which I'd be happy to talk about afterwards. A few surprising observations that survival did not correlate with age of diagnosis. A second surprising thing is that women actually survive better than men. So maybe they're more aware of their bodies. They were diagnosed earlier. No, they're diagnosed at the same age, the same stage. We're wondering, could it be hormones? We're exploring, could estrogen be protective, testosterone inflammatory? Or maybe it's a double dose of some genes in the X chromosome, or possibly loss of a tumor suppressor on the X chromosome, which in men you lose one, you're in trouble. Another strange observation is that survival did not correlate with tumor size, nor did metastasis correlate with tumor size. So there are some patients who had tumors that are 20, 25 centimeters who had no metastases, and other patients who had tumors of half a centimeter where they had metastases. And we're actually finding the patients seem to fall into two categories, those that are highly metastatic, those that aren't, but when we compare the tumors to each other, we don't see any differences. And we're beginning to wonder, maybe there's something in the patient background that's there. So survival correlates with whether you have metastases, whether you have extrahepatic involvement or the number of tumors in the liver. So this is similar, for example, to cases of melanoma, where patients who have ApoE2 have much more metastatic tumors, whereas ApoE4 don't. And we're now doing a GWAS study to see, can we try to figure out what's different about their background? So what I hope I've shown is that fibrolamellar is the result of somatic mutation. It's not genetically inherited. All cases of fibrolamellar are a result of dysregulation of protein kinase A, with the vast majority of them being this 400 kilobase deletion in chromosome 19. This fuses a heat shock protein with the catalytic subunit protein kinase A. This produces a fusion gene, a fusion protein, which is sufficient to recreate fibrolamellar in the livers of mice. Drugs in the clinic have no effect on fibrolamellar. Drugs that antagonize the oncogenes that are regulated in fibrolamellar without efficacy. A functional precision medicine screen, though, does identify a number of agents that show efficacy on the cells dissociated from tumors in mice, cells dissociated directly from tumors in patients, tumors growing in mice, tumors in patients. And we can understand the efficacy of these drugs from analysis of the biology of the tumor. I want to thank the people who gave us support. A lot of this started both with private foundations who jump-started it, as well as Gunter Blobel, who very generously supported some of the early work. Since then, the NIH has been incredibly supportive, as well as other people. I want to thank all the members of my lab who have basically sort of redirecting the research now to fully work on this project. They've asked me to show the following slide to give you the impression that they actually go out and they have fun. This is not true. This is photoshopped, but this is only because they want me to tell you their postdoctoral positions available, and they want other people to come and join them in their misery. And then finally, above all, I want to thank the fibrolamellar patients. They're really the vanguard of the battle. They've given us tissue samples, blood samples. They've recruited other patients. They've done a lot of the fundraising, and all of these have worked in the lab. I really owe everything to them. So thank you very much.
Thank you. Thank you. That was a fascinating talk. Regarding the photoshopping, we're going to start about the science in a second. I have another question. I saw there was at least 2 or 3 slides with a dog. That dog was never mentioned in your acknowledgement. Did I see this correct? Do you have a dog in the lab?
2.
Okay. All right. Okay. So I guess I was not wrong about this. Okay. So let's get now become serious again. And, so I'm going to ask, there are some online questions, and then I'm going to have the people that put on the camera ask the question. So there is one from Alison Cook. As far as you know, is there some adaptive function of the chimeric kinase that is evading homeostatic control in these cancer patients, or may there be?
Good question. Since it's not being shut down in excess, so it turns out there's an excess of this catalytic subunit over the regulatory subunit. I would have imagined as you increase the catalytic subunit, you'd also be increasing the regulatory subunit. But in fact, the catalytic subunit is in excess. So somehow it seems to be, actually, just as a background for people who aren't familiar with protein kinase A, it turns out that every catalytic subunit ends up being in a complex with the regulatory subunit, which in turn binds onto certain A kinase associated proteins that help localize it. And the regulatory subunit inhibits it until such a signal comes to let go. The catalytic subunit is free and active. Much to my surprise, I would have thought the regulatory subunits be increased. Somehow it's escaped this homeostatic control. Allison, is that what you were driving at? Allison?
Yeah, that's what I'm driving at. So I'm just I'm trying to kind of back up, and because I'm thinking that there is often some kind of functional precursor that leads to an epigenetic change that is precancerous, or this is my current thinking right now. And your research tends to kind of conform to that. So I guess I'm wondering if there's some use in the liver for this chimera under normal conditions, but that the body is just kind of going berserk, like doing it way too much and escaping control. And that's leading to the cancerous, precancerous situation. So that's kind of my general thinking. Thank you.
Yeah, so protein kinase A plays an incredibly important role in all different aspects of liver physiology. It's there, it's very active. What we found is that in the tumor, and only in the tumor, on one you have the normal kinase, but there's on the second chromosome this weird fusion that occurs that both increases the expression of the kinase, but also slightly alters its activity, what it's acting on. And those two seem to be sufficient to produce the tumor, that and nothing else. And the reason why I'm saying it's sufficient is that we've now gone into a few different mouse cell lines, and actually another group that was on the slide. They've done this yet in another mouse line, just engineering in or just expressing that one fusion and nothing else is enough to give you the tumor. So I really think that's all you need. And I think the difference is due to both the increased activity of protein kinase A, which alters some of the physiology of the liver, but also that altered activity. It's now phosphorylating a few novel phosphosites that's what I think. So in that sense, I think it's different from a lot of other tumors.
Okay, thank you.
Aitan, you want to go next?
Oh no, I just shown my face out of courtesy. I just want to say it's a fantastic talk and fantastic work. Really, it's so commendable, and to see all this volume of work, just amazing. And thank you so much. My interest is that, we looked a bit, we talked a bit in the past, you probably don't remember. We are looking at applying synthetic lethality to predict immunotherapy response into FLC. We have a few cases. We actually have a case report coming up in GITC soon. So of course, these are, and we have a few success in predicting who will respond to immunotherapy, but very small numbers. But your work is just amazing. Thank you so much.
We've tried a number. Thank you very much. If you can come up with some prediction, that'd be great. We tried a number of immunological approaches. What you didn't see the talk was all the different rabbit holes we went down that just failed, or if they didn't fail, we got to a certain point where we decided it wasn't worth pursuing further, and we turned around and went back. And what we didn't know, and actually this is important for any graduate students or post-bacs who are there, when you turn around, you never know if you're turning around and you were still like just, a short distance away from the ending, or in fact, it wasn't going to come up with anything. One of those was immunological approaches. So it turns out there's only a small percentage of patients who actually present a peptide from the fusion junction on the HLA-1 or 2.
So our approach is a different one. I'll just send you, it's a very short
Sure.
paper, you'll see.
If you get something to work, great, even on a subset of patients.
Thank you.
Okay, so Mike Gottesman.
Sandy, first of all, hello. It's been a while.
Oh, hi, how are you?
Let me go back to your answer to the first question about the regulatory subunits of cyclic AMP dependent protein kinase, which normally in cells are in excess and bind all of the catalytic subunit and direct that catalytic subunit, and in the presence of cyclic AMP, catalytic subunit is released. You mentioned, and I make sure I got this right, that in your chimera, the regulatory subunit doesn't bind. Is that true, or?
No, if I said that, I misspoke. What we did is we calibrated, we did 4 or 5 different things. We calibrated the amount of catalytic subunit, wild type, catalytic subunit regulatory, catalytic subunit, wild type, catalytic subunit fusion, and the regulatory subunits. We did it by doing mass spec in which we doped in set amounts of peptides. And we did it to calibrate how much we had of each. So observation 1, there's an excess of catalytic subunit in the tumor compared to regulatory subunit, because we can actually calibrate the absolute numbers. That's experiment 1. Experiment 2, if you just measure free PK activity in the cells, it's much, much higher in the fibrolamellar cells. Experiment 3, if you come in and you just pull out all the arch subunits, you do it in the normal tissue, you pull out all the catalytic subunit. You do it in the fibrolamellar, you've got a lot of excess catalytic subunit. The next test, you pull out the free catalytic subunit, you get almost nothing from normal liver, you get a lot from fibrolamellar. So I think there's a lot of free catalytic activity. It's not that it doesn't bind, it's just it's increased in the protein level.
So, and it's not regulated, basically.
1, it's not regulated, and 2, its target is slightly altered. So, and if you just overexpress PRKCA, you can get a lot of the phenotype of fibrolamellar. So for example, ornithine aminotransferase that goes up, it goes up just from overexpressing PRKCA. You just don't get transformation.
Right. So just to remind people that in most epithelial cells, cyclic AMP dependent protein kinase stimulates growth of cells. And you may remember many years ago, Howard Green was trying to grow skin cells in tissue culture, and he added cholera toxin, which stimulates cyclic AMP protein kinase, and got beautiful growth of cells. So not surprising that an excess of cyclic AMP dependent protein kinase will stimulate growth.
Mm-hmm. Good to see you again.
Yeah, great.
So yeah, Sandy, I certainly shared all the sentiment from everyone. I always enjoyed your super energy, and particularly the lesson that we could actually learn from intramural side, really study the real disease. What one disease you can actually really make such an impact. Now, having said that, I think it was also peculiar to me for this disease that you have one single dominant driver mutations. Yet you have such a complex phenotype. Many of the phenotype that we know from the hepatocellular carcinoma, for example, small tumor could be extremely aggressive, larger tumor could be very relatively benign. At the same time, male tend to be more aggressive than female. Those are all phenotype that we know from the other cancer type, hepatocellular carcinoma, one example. But you have the single genetic dominant driver as compared with liver cancer, which has had really heterogeneous driver. I just wondering what are your thoughts on this particular driver? The discussion that you have also had a real look at the catalytic subunit of this particular oncogenes, and whether there are some subtle difference between different individuals that somehow transmit the phenotype. But this is somehow against what we know about the human tumors, because we always think that there's a multiple genetic alteration rather than single lesion. So it must be the heterogeneous features that are driving by lots of other things. So what's your thoughts on?
So it may be there are just different classifications of cancers and tumors. For example, in Ewing sarcoma, or when you've got BCR-ABL or rhabdomyosarcoma, it may be a lot of these childhood cancers may have a single unique driver. which just puts them in just a different disease class from, let's say, some HCC where, if the patient has a lot of cirrhosis, they've damage from whether it's viruses or alcohol, they could have many different genetic aberrations, and it just be sort of a different class of disease.
All right, so let me just go back here to this chat and just ask, there's from Bryce Wilson. He mentioned this is a Tour de France. Is there something unique about the liver that makes them susceptible to this chimera? For example, if you put the fusion into a non-tumorigenic prostate line, would it become more aggressive?
We haven't done that. It's actually, it's one of the many questions we've agonized over. We have tried generating mice where we can use Cree to induce it everywhere, and we were not getting tumors elsewhere. And we're trying to figure out why. So my initial hypothesis, which clearly has some problems, was that maybe these two genes, actually, can you see my hands? I don't know if that's in the video.
Yes.
Maybe these two genes are right near each other in the liver. And in fact, if you do high C, we can show them near each other. And maybe in the liver, they're close to each other. Maybe other organs aren't. And when there's some insult that comes in, that's why you get the break in fusion. And maybe they're not near each other in other organs. But that wouldn't explain why, if we turn it on with Cree in other organs, it doesn't happen. We've started now trying to put the fusion kinase into cytosol of different organs and ask what gets phosphorylated in each. What are the unique extra phosphosites? And could that give us some insight? And that's a rabbit hole we're still going down. I'm not giving it as high a priority, in part because my immediate focus is to come up with both therapeutics and some blood and urine-based diagnostics for patients. Well, we're going after the pathogenesis in the hope that it will give us, some new therapeutic targets. But actually, the immediate goal is to get a therapeutic into the clinic, and then we're continuing to pursue it. I just don't have a satisfactory answer.
So talking about the therapeutics, if I remember correctly, you showed some data for the in vitro screens, and that if I saw this correctly, there seems to be a difference in response if you look at primary tumors versus metastasis. You had like 5 different cell lines,
Yeah.
I think, and they said liver or metastasis, if I remember this correctly. Is there indeed a difference between susceptibility or how they respond?
No, that was. Hold this up here. So early on, we had these 5 where actually these
Yes.
were mets, these were primary. Now that we have about 18 of them, there doesn't seem to be a consistent difference. And actually now we've done the transcriptome, we've done whole exome between. I think between so students writing on a paper now, if you look at all 65,000 transcripts, there are less than 10 that differ in the mets from the primary. And I think this just may have been a question of just growth over time.
Okay. All right. So
We were hoping to see that.
let me thank you again. That was really an amazing talk. I personally have to say, I learned a lot, and very much enjoyed talking to you and listening to you in the past hour. And with this, I'm going to close and ask Anu, because I should know, but I don't know when we are going to have the next speaker.
Our next speaker is on December 1st. It'll be Matthias Heichenwalder.
Oh,
It'll be at 9 a.m. on December 1st.
okay. Thanks, everybody.
So I go back into